Introduce Dr. Karin Bjorkman, who did her PhD at Stockholm University in zoology and aquatic ecology, which she finished in 1999. And since 2001, she is a research specialist at the University of Hawaii and has conducted research at Station ALOHA. So today she will talk about phosphorus cycling in the North Pacific Gyre. Welcome.
Thank you. I know I'm not much of uploading any information on the web, so it's really hard to find out anything about me. I apologize about that.
I actually wanted to ask if you also did your master studies and bachelor studies at Stockholm University.
I did actually, as a matter of fact, in the same discipline. So yeah, I was spent a lot of time at the Stockholm University and a lot of time at the Department of Botany as well. At one point, I thought I'd become a taxonomist in botany, but then I changed gears and became something completely different, more of an oceanographer, microbial ecologist. Okay, so I think this talk is approximately 30 minutes long, and I wanted to give more like an expose of the phosphorus in an oligotrophic marine system where I worked most of my life, and first give you a little introduction on when phosphorus was discovered and why phosphorus is such a linchpin in all living matter. It's so important for life. And then going over a little bit on the global marine phosphorus cycling and some terminology that we usually swing around and it becomes completely incomprehensible if you don't have any sense for that. And then dive into what we know about phosphorus at Station ALOHA. This is just like a little snapshot of what's going on there. So there's so much more you can talk about, but that would be several lectures if that would be. So it's trying to be just little bits and pieces here and there. But let's start at late 1600s when phosphorus was actually discovered. And it was discovered by a man named Henning Brandt. And he was an alchemist and basically spent his days in the lab trying to, as alchemists do, right, try to turn base metals into gold. And he wasn't very successful. And I think he actually pretty much bankrupted his first wife and then managed to marry a wealthy widow so he can continue his passions here as an alchemist. And this picture here shows you the night of discovery in his lab. And he has this flask where he has distilled down a lot of urine into very small volume. And as you know, maybe you know this from if you're an alchemist, you try to make something into gold. So you usually start with something that looks like gold. So what is better than a bunch of pee? And here he has distilled this down into something. And all of a sudden you have these white fumes coming out of the flask. And it leaps out into the floor and it catches on fire. And after this all calms down, he realizes he has this white weird residue in his flask and it's glowing in the dark by itself. So he realizes he has actually discovered something that's unique. Even if it may not be gold, it's very different and it's very interesting. And he names it phosphorus. And that basically means light bearer or light bringer. And it was actually almost 100 years later when the founder of modern chemistry, Antoine Lavoisier, discovered that phosphorus was actually an element and not just the substance of some sort of combined material. It was actually an element. And it's often called the devil's element because it was the 13th element that was discovered. It also has this eerie glow in the dark, right? So it has this light emission thing going on. And later on, they used this capacity of phosphorus to actually almost self-ignite and to make matches. So part of these match heads was actually phosphorus or elemental phosphorus. And they were called, we also call them Lucifer sticks. Also again, Lucifer is also again, light bringers, the same thing, right? That fallen angel and has to do with light. And you may have heard about matchstick girls and matchstick workers that produce these matches, the ones you can strike on any surface and they will ignite. They sometimes came up with this so-called phossy jaw, which is a nasty, nasty kind of disease. And it has to do with the fumes of phosphorus that actually dissolves the bones in your jaw. So that was a really nasty thing that a lot of the workers that worked in the match factories happened to them. You can look it up on the web, it's really nasty. But I'm not going to show you pictures of it. And other things that makes phosphorus kind of nasty is obviously it's been used in nerve gases and pesticides and a lot of other things because it's so central to life. When it links into wrong things, it can stop processes in life. So in spite of all this, phosphorus is really, really the staff of life. It is some unique properties of phosphorus that makes it central to all life. And part of that is that it in nature occurs like phosphate or a trivalent acid that has 3 binding sites and a lot of negative charges. And that turns out to be really, really important for life. So we know, for instance, for structural components, you have phospholipids, you make bilayers of phospholipids, so you can make compartments and cells. So that's kind of because that's a hydrophilic point of that with all these charges on the outside. In terms of energetics, such as ATP, which is an anhydride bond with 3 phosphate units, is involved in almost all energy transactions in the cell. It uses the high energetic bonds that is between the phosphate units. And the interesting thing with that is that it shouldn't be stable thermodynamically, but it is because of all those negative charges that supports this 3 bonds across that thing. And obviously for life, it's central in genetic material, in the DNA and RNA, which it kind of links is the, what do you call it? The ester link between the nucleotides. You can kind of put them on and they're easy bonds to break and put them together again. And again, you have additional negative charges that will stabilize these structures. So it's really, really important. And nature picked phosphorus because of these chemical properties that it has. So to me, this is a very central part of the periodic table. So if we look at where we can find phosphorus in the world, it's really a simplified scheme here that I took from a very old ecology book. And you can basically divide up the whole world in 4 different compartments. And it's solid earth, the water components such as oceans, lakes, and rivers and stuff like that, or the atmosphere, or bound up in biomass in different sorts. In terms of phosphorus, the atmospheric component is very, very small. Essentially, the only parts that will be in the atmosphere is in the form of dust particles that have been kind of blown off the continents and travel like that. It doesn't really have a long-lived gas phase or anything like that. This is different from nitrogen, for example, that has a very, very large pool in the atmosphere. And then these phosphorus will cycle between these different compartments. From the solid earth, it would be eroding into the waterways where it can be taken up into various life forms, etc., and it circles around. But just keep in mind that this atmospheric pool is very little or very small. So the central part will be around the solid earth, life and water. And in terms of where you find phosphorus reservoirs, almost all phosphorus is bound up in solid earth. So 99.98% is in solid earth. And that erodes and kind of replenishes these other pools. So those 0.2%, almost everything will be in the deep ocean, where it's away from the sunlit upper ocean, where you have most of the productivity. So we have no atmospheric input. And if you're in the ocean, almost all new phosphorus will have to come in from below. And I'd also like to point out the difference here in what's bound up in land plants and animals versus what's bound up in ocean lands, plants, and animals is very, very different. And it tells us something about how fast these pools are turning over. So land biomass is old and long-lived, whereas in the ocean, things turn over pretty quickly. And you can view this different time scales and different spatial scales, obviously. So if we look at the erosion time scales and the big rocks going underneath and everything, we're looking at millions of years and very, very large time scales. And with the continuum down to this pink box where I spend most of my life, which is short time scales and small distances. That's where we look at what happens with plankton productivity, stoichiometry of various elements, what happens in the microbial world, basically. So before I go on, I was going to do a little bit of phosphorus terminology. It's not going to be too complicated, but we can view this upper ocean pool of phosphorus, as we frequently called TDP, which is the total dissolved phosphorus pool. That's containing all the inorganic phosphorus, and it contains all the combined molecules that contains phosphorus. The thing to remember is that we limited what we have in our toolbox and what we can measure. So even though we may want to measure all the different components in here, our tools doesn't really allow us to do that. We can measure this and call it the total pool, but maybe not the separate components within it. Another part of this is what we usually call soluble reactive phosphorus, which is, again, just because this is what our tools allows us to measure. And that is represented often for the inorganic pool, which is directly available to bacteria and phytoplankton. They can take what the inorganic phosphorus that we measure here is directly available. What's remaining in this other pool is probably available to a certain degree, but maybe not all of it, and it will be in different time scales. But you do have exchange between these pools. But if you, like me, yeah, I'm a biologist by training, even though I ended up in oceanography and more of chemistry at least kind of part of my life. What we really want to know is obviously what portion of this is it that the microbes out there are really using. And we usually call that the bioavailable phosphorus pool, or BAP. So when you hear these kind of terms later in my talk, it kind of represents different parts of this total pool. And the SRP pool and the total pool we can measure directly through chemical analysis. Whereas this other pool that we really want to know, you can't get to unless you do bioassays. And I will show you later in this talk a way of getting to this at the very end. And then this phosphorus obviously cycles through the upper ocean. This is the euphotic zone where you have sufficient light for primary producers to make more carbon or fix more carbon. We have a small arrow in here from the atmospheric input, which I said was dust deposition, which is very, very small and really insignificant in terms of phosphorus. So most of the phosphorus has to come in from below somehow. And if you're out in a system like the North Pacific Subtropical Gyre, most of this phosphorus here is going to be recycled and recycled many, many times. It's a recycling intense system. Little comes in here, little goes out, and most of it is maintained here and cycles around. And it's cycled through microbes, obviously. As I said before, this SRP pool, which is the inorganic phosphorus pool, can be taken up directly by the microorganisms. They just can transport it straight through their transporters into their cells. Whereas portions of this other pool will have to be broken down before you can access the phosphorus. There's very few phosphorus compounds that can be transported directly into the cell. So that, oops, what happened there? Okay, back. So that is something to keep in mind too. If you have a system like out in this ocean where these pools are actually really small, and this pool is much smaller than this pool, it should be advantageous to be able to utilize the phosphorus that's locked into this much larger pool. But it comes with the cost, of course. So if we now jump on to Station ALOHA and the time series, and I don't know how much you've heard about Station ALOHA and the time series. I've obviously lived with it for 25 years. So for me, it's home turf, and I don't know how widespread the knowledge is. But anyways, here we have the North Pacific Subtropical Gyre, which is, the largest continuous biome on our planet. It's a very, very large ecosystem. And it's obviously important to try to understand what processes govern these type of systems. Because even if all the rates are low and slow, small changes can actually have large global effects just because the ecosystems are so large. And Station ALOHA, which is obviously, of course, I should say, an acronym, because that's what we always do. It stands for a long-term oligotrophic habitat assessment. And it was founded as part of the Joint Global Ocean Flux studies in the late 80s. And the whole purpose of it was an international project. And there were several different countries involved. And they founded different stations around the world. And Station ALOHA was representative for the North Pacific Subtropical Gyre was the idea. And it was really to get the better understanding for the carbon flow through the atmosphere and oceans and how biology influenced these different cycles of carbon. And also how carbon can be removed from the atmosphere and sequestered into the ocean and shuttled down to the deep sea and basically be removed from the upper ocean. Because back in the 80s, obviously, we started to get the understanding of these cycles were very important to get a good understanding for, but the knowledge just wasn't there. So Station ALOHA is situated about 100 kilometers, 10 mil, north on this point, Kahuku Point of the island of Oahu. And the reason it's situated here is basically, as I said, we're trying to get something that is representative for the much larger gyre system. So it's a deep ocean site. It's almost 5 kilometers deep. And it's really typical for an oligotrophic ocean station. There's very low nutrient concentrations in the upper ocean, very low standing stocks of phytoplankton and chlorophyll. So it's a blue water station, which means that you have little particles in the water. So light can penetrate very, very deeply into the ocean, in the water column. So you have light down to probably 150 meters or so. That's sufficient for phytoplankton to grow. And I'm sure you heard about Prochlorococcus. It's the dominant phytoplankton out there, both in abundance and in biomass. But when this program was founded, or when people started thinking about this, we didn't even know that Prochlorococcus existed. It had yet to be discovered. So there's a lot of things that happens. 30 years sounds long to you, but we're still at that edge of discovery as long as we go out and look at things. And so a lot of the planning for these, what we're going to do out here, was put in place without even knowing about this really, really important microorganism out there. And this is a research vessel, the Kila Moana, that lots of us people have been out on. So to show you a little bit about the upper ocean biology and nutrient structure at Station ALOHA, this is very typical for what you would see at an open ocean station in a subtropical gyre, where you have very low nutrients, lots of lights. So we can see from the surface, this is just the top 200 meters, but you can see that the light attenuates down with that, here with this curve. Two of the important nutrients, which is here SRP, as I talked about, the inorganic phosphorus pool, and the nitrate pool, two building blocks of biomass, are really, really low in the upper ocean, until you get down to about 100 and 125 meters, when they started to pick up a little bit again. Chlorophyll in the upper ocean is also really, really low, until you get down here again to about 125 meters or so. And this is what we tend to call the deep chlorophyll maximum, which you almost always see in these type of environments. Interestingly enough, if you look at productivity instead, primary productivity, these guys here, the chlorophyll bearing cells, it's always highest in the upper ocean and then declines. And this is a function of the amount of light and the amount of biomass. That these guys have low chlorophyll has a lot to do with that there is plenty of light. They don't need to have a lot of chlorophyll to go as fast as they can anyways. Whereas if you get down here deeper into the ocean, the light is so low, so you pack on a bunch of chlorophyll to really strip out that little bit of light you have to build more biomass. But it isn't more biomass down here. So it's a little deceptive that way. But it's this is a very typical what you would see in these type of environments. So when I first got out there a long time ago, we had a lot of questions about phosphorus because we knew that it was low. We didn't know how it varied in time and space. We didn't know if different pools varied in different spaces. We knew very little about how fast the phosphorus was turning over, how fast the microbes would take it up. And we knew basically nothing about how much of this dissolved organic pool they could utilize, which seems like they should if it's very low. And then doesn't matter who you are. The different groups of organisms utilize phosphorus differently or at different rates. So if I just start with this first question. So this is a time series. It's almost the whole time series. It's missing a few years at the end. It's showing contour plot of the top 100 meters. And it's obvious, right, just looking at this, that it's a lot of variability over time. Over depth as well, but especially over time. We have periods of relatively high concentrations followed by periods of very low concentrations. And overall, we're talking about nanomolar here. I hope that tells you something, but that is low, low concentrations. And this also, I think I want to just point out that having a long time series like this allows you to actually try to tease apart what's really happening, not just on annual time scales, but maybe on decadal time scales as well. Because you need a lot of years to actually tease out the variability that you have and what matters. Case in point, this is the same thing, but we're just looking at it differently by integrating that phosphate pool that we had in the top 100 meters. We were super excited when we saw from these first 5 or so years that, oh my God, phosphorus is running out. Oops, phosphorus is running out. It's just going down. It's going to disappear. I mean, we lost, what is this? 70% of our phosphorus pool over 5 years. And then we had this influx again and a drawdown. And then it did this. And then we had this massive influx again. So we had written a very exciting paper here. We wrote another exciting paper here. And then it was like, what is going on? So that I should actually, we have figured out kind of what drives these dynamics. And it's very complicated. And it's really a global phenomenon that drives these changes in this system. It's not just what the microbes do, it's the whole climate system that affects what we see out here.
Excuse me?
Yes.
Can you give us an example?
What yes, do you really? Okay, I can give you an example. So there is several ocean indices that affects the North Pacific tropical gyres, that affects the circulation rate of the gyre. And a lot of that has to do with, if you have heard about the Aleutian low, maybe. The Aleutian low sits over the Aleutian Islands, basically. And that moves on decadal timescales up and down. And that affects the entire gyre circulation. And it also affects some of how the jet streams go. So what we think we have figured out is that during certain periods of time, this place, depending on how these climate forcings are forcing the gyre, we get more or less input of atmospheric ion that also comes with dust from China. And if it comes more south, we will actually stimulate a lot of nitrogen fixation. And when we have nitrogen fixation, we add nitrogen to the system, but we can't add any more phosphorus because that comes only from below. So when we have ion input, we get nitrogen fixation that will draw down the phosphorus pool. And when that lets up again, it allows the phosphate to build up in here because we no longer have that extra nitrogen fixation coming in. And we don't have any nitrogen really coming in more from below either. So that is usually stripped out and we have a reservoir, kind of a reserve of phosphorus that can't be drawn down because we don't have the nitrogen fixation going. Does that maybe answer? It's a really cool story. It's a recent paper that came out just last year that has a really nice explanation for this. But I wanted to show you some of the other stuff. And that is more on the microbial side, what we found out of how fast these guys are taking up phosphorus. And because I say this is very near and dear to my heart, because this is, I want to say, my tool chest and my play box is using radioisotopes to try to tease these things apart. So I worked with a lot of different radioisotopes, but particularly phosphorus 32 or 33. And the cool thing with this is that you can add these compounds, or it's actually phosphate as phosphate, at super low amounts. So you don't affect the existing pools. You don't actually add phosphorus to them. So they are true tracers. And then you can, you know how much you added, and then you can follow around where it goes, and you can get how long it takes for these pools to turn over, and you can calculate how fast this community is taking this up. And then, as I said, you can also look into bioavailability of some known compounds I'm going to show you. And finally, you can use these same tools for try to figuring out this bioavailable phosphorus pool via some bioassays. Yeah, I spent a lot of times using radioisotopes, and I think it's fantastic tools. So this is just a picture from the Pacific Ocean when it's not so Pacific. So one of the things we wanted to know is obviously how long does it take for the phosphate pools to turn over depending on the concentrations. So one of those things we found, these are all experiments that were done with whatever the concentration was out in the field. So these are not added. The phosphate here is not added. This is what we measured at these different occasions. And then we looked at how long it took for that amount of phosphorus to turn over. And as you can see, it's a pretty good relationship between the turnover time and the amount of phosphorus, right, in the water. So that tells us something about what's going on, which we didn't at first think we would see. But what that means is really, if you convert this to uptake rate instead, we have no correlation between this. And or very low correlation anyways. And part of that we thought is this Prochlorococcus community is really not phosphorus stressed in almost any concentration that we normally see. So it's well adapted. And another way of seeing this is just adding a bunch more cruises and a bunch more data points. And again, look at the turnover time and the concentrations we measured in the field. And you can see it's kind of like a swishy type of relationship. And what it means is really, here's the mean concentration over those many years. It's about 50 nanomolars of phosphate. When you go below this curve flattens out. And what that really means is that for any additional phosphorus that you add down here, nothing happens to the turnover time, which means that the rates are going up. It means that down here, they are phosphorus limited. So any extra additional phosphorus comes in means that you're going to take it up faster. Whereas when you come out here, it turns into a linear relationship, which then mean that any additional phosphorus that you're adding doesn't change the rate of uptake. They're already going as fast as they can. And all it does is add to how long it takes to turn this pool over. And we have measured, as I said, this is the mean concentration at station ALOHA. And we've done kinetic studies, and I'm going to show, I think, the next slide, a little bit of that this Prochlorococcus community have saturation constant. If you look at those kind of studies, is around 25 to 30 nanomolar. So it's lower than the mean. And it also says that Prochlorococcus is really uniquely adapted to these low nutrient environments and does really, really well. So this is how I established that Km value, or half saturation constant, when you do other types of experiments where we actually added a bunch of phosphorus to a background concentration and then looked at the uptake rate. So all of these same color dots are the same experiment, but at different concentrations of added phosphorus. So one of the things when you're out there, obviously, you can't pick where you start. It's just going to be what it is. And then you add more. And a lot of these experiments, you don't see any rate changes or anything. It's just flat, which means we're above that threshold where they are already going as fast as they can. Whereas if you come out here when it's really low, you get the really nice kinetic relationship, which again means that the more you add, the faster they will take it up. And this star here is probably 30 experiments from station ALOHA, and the average what we saw out there. So that again comes down to, you know, the mean concentration is about 50, and this is the rate, is about 3 or 4 nanomolars of phosphorus per day, liter in day. So it's not fast. It takes days to weeks to turn this pool over, what they have there. Another way of looking at that, when we were interested in seeing if different components of the microbial community would react differently to additional phosphate that we also added. We happened pretty lucky and was really low when we started. So these are the actual background concentrations. This is not what we added. This is what we measured. On the relative scale, we can see that the smallest group that was between 2 and 2 micron, which would contain the Prochlorococcus community, but not really any of the larger diatoms or anything like that. Almost all the phosphorus will go into the smallest cells. And it wasn't until we got up to about 75 to 100 nanomolars phosphorus that we saw that these larger cells starting to be competitive for the phosphorus. I'm viewing this another way. I know this is a little ugly slide. If you're looking at 3 different communities here, this is the Prochlorococcus community. Saturates at the really low concentration of phosphorus. So we have this half saturation constant of about 20 to 30. If you then look at trichodesmian colonies, they need more phosphorus before they start being competitive. They need to have a higher concentration before they start to take it up at any significant rates. And diatoms are even higher. So that means within this little sweet spot back here, which is the typical station ALOHA community concentrations, Prochlorococcus is the most competitive organism. You have to have a little higher before the diatoms can really, and trichodesmian can really kick in and start being competitive for the spool. Okay, now about bioavailability of, how much time do I have? Of the dissolved organic pool. So you can view this, obviously, as I said before, that depending, you can view maybe as a spectrum of different compounds being more or less available. And depending on how stressed the community is, you're going to dig into different compartments of these DOP pools. And this, when I say DOP is like a blanket statement, it's a very diverse, chemically diverse pool. And we don't really know what all of these compounds are and how they are available. We're learning, I should say that. But this is what we know. Chemically diverse, poorly characterized, and it's much larger than the inorganic or SRP pool. So when you're nutrient stressed or phosphorus stressed, if you can tap into that, this should really be helpful. So we devised a really simple experiment. Other people have done similar stuff. If you want to compare a known phosphorus organic compound to inorganic phosphorus and see how much of that unknown compound, or known compound, I should say, is competing with the phosphate, we basically did an experiment where we had a control here, a negative control, where we hadn't added anything. It was just seawater and our tracer. And then you had a known amount of inorganic phosphorus that kind of dilutes your label. And then you had the compound that you wanted to test, how available it is. So you're looking at the difference between the positive control, that is inorganic phosphorus or nothing, and this compound. So what it essentially means is that this is not anything complicated. What it essentially means is that, maybe I should go back here, it's easier. If a compound is as available as inorganic phosphorus, this red line will be overlapping the blue line. If none of the treatment here, the DOP compound, is available, it will be equal to the non-amended controls. So it's basically dilution. You dilute your tracer with either the inorganic phosphorus or phosphorus coming from your known compound. So we call that the bioavailability factor, where 0 is when it's not available and 1 is it looks exactly like inorganic phosphorus. And then we tested a bunch of different compounds. A bunch of sugar esters, and then it was nucleotides and some inorganic polyphosphates, for instance. And what we found was, over short time scales, like hours, none of them are particularly bioavailable. It's like a few percent will be taken up compared to what the inorganic phosphorus is. But the nucleotides stood out as a group that at least was more bioavailable than anything else. And as I said, it's important to remember that short time scales, over longer time scales, they're probably all being degraded. But immediately available, this group is the important one. So then finally, I wanted to show you a little bit about this bioavailability assay that we can do. And again, we come back to some really clever things. I wasn't the clever one who devised this, but I did the experiments. You use the radio tracer, and what you're looking at is the specific labeling of the internal ATP pool. This is really complicated, but it's really brilliant. So what you do is you put your tracer outside here, and you look again at this dilution of your tracer that you measure. So you have a known amount of your inorganic type, and you have some amount here of the organic pool. Inside here, this is the clever part. Almost all of the phosphorus that comes into the cell will label the ATP pool, because ATP cycles very, very rapidly inside the cells. So this pool will be uniformly labeled with your tracer with very short periods of time. So that's the advantage. And then we can measure this, the content of this, and we can see how much of that actually have our tracer. And that amount should match how much phosphorus has come from different sources. So we can, see if this is a lot more here, this is more dilute than what this would be, it means that some of this has to come from the DOP pool. So what we found was essentially that when you look at the bioavailable pool, it was almost double that we measured as the SRP pool. And that obviously affects when we calculate our uptake rates from our turnover time and our measured concentrations, that the rates based on the bioavailability pool is much higher than the other ones. But the most important part is that almost half of the phosphorus that the cells out there used came from the DOP pool. This doesn't mean that it's a large percentage of the DOP pool, because it's not. But the cells use almost 50-50 for their phosphorus needs will come from the dissolved organic phosphorus pool. All right, so I think I'm about on time. So I hope you had appreciation for how important phosphorus is as the staff of life. And it's really central to many, many functions of the cell. And also a little bit about the phosphorus cycling and dynamics in the North Pacific subtropical gyre. I really do want to point out, I think that time series work and data are very important for understanding on global processes. And they become almost more important the longer they run. And there are a few stations that are as old as station ALOHA, and the other one is the Bermuda Atlantic time series as well, BATS. They're sister programs, they've gone on for the same amount of time. And I think that's really important for our understanding of, if not else, the climate change and all those things. These time series stations are super important. And I show you these are things that, you know, the inventories change over time. They're very much more dynamic than we thought they were. Originally, the view of the subtropical gyres were that basically were dead. There were very little going on. They were static. And we now know that they're really, really dynamic and productive. What can I say more? Yeah, obviously Prochlorococcus is the important organism, and it's normally not, I would say normally it is not stressed at the concentration that we see out there. It's not limited, but the larger cells out there, like diatoms, probably trichodesmin, other important groups will feel a phosphorus stress much earlier than Prochlorococcus. And as we saw, that DOP can provide about half of the phosphorus needs for this community. So I think that is, this is it, folks. So let's see, stop sharing. All right, so this should be some chat. Oh, bunch of chat.